Stanford University.
Thank you. Thank you. So thank you very much, Bill, and congratulations to you and your colleagues on this amazing achievement. I know that creating cross-school, cross-campus, interdisciplinary efforts is no small effort. You're hiding your black and blue marks well, but it really is great. And in some ways, the whole idea of the Broad Institute is to do the same, bringing together actually something that was more challenging, which was so you can take over some new aspirations, but bringing together Harvard and MIT, not always friendly with each other, and the Harvard hospitals to create a space for interdisciplinary collaboration grounded in technology. So very much in the kind of spirit of what you're developing here. So I'm going to talk about translational psychiatry, and I'm going to give you more of an overview of what we're doing at the Stanley Center, strategy, some results, but instead of going very deep in one area, I think it's worth describing how I and my colleagues are thinking about the problems facing us. Bill reminded me that this audience has very diverse backgrounds, so I'm not going to spend a lot of time on introduction, but I think this one slide makes a useful point, which is this is from the Global Burden of Disease study, and the Global Burden of Disease study was initiated actually by the World Bank with the World Health Organization to give advice. If any of you were the health minister of a country, where would you spend your marginal dollar? That is, what diseases where treatments existed are creating the greatest burden in terms of either premature mortality or years of healthy life lost to disability. Now, the neuropsychiatric disorders do cause excess mortality. There are between 800,000 and 1 million suicides globally per year. And people with schizophrenia actually have a decrement in life expectancy of about 20 years. Part of it because they don't access health care. Many of them are smokers, but there are other issues that we probably don't understand. But the key issue for diseases like autism, schizophrenia, bipolar disorder, OCD, and so forth, is they cause disability. So in this graph, this is basically ages from, I know you can't see the x-axis, from 1 to 85. And this is the contribution of mental disorders to years of life lost to disability. And what you can see, and here below it, you can see the neurologic disorders getting more important with age. This is, Alzheimer's, Parkinson's. So the take-home point here is that the neuropsychiatric disorders are the severe impairing chronic diseases of the young. And as a result, they impair the ability of kids to learn in school, of people to work, of people to form families, and they create an enormous societal burden. Unfortunately, therapy has been very much stuck for more than half a century. Now, there have been advances in psychotherapies. There are some interesting cognitive remediation therapies that are proving very helpful in prodromal schizophrenia. They're very intensive early behavioral therapies that are very useful in autism. And for the non-Cartesians among you, it's not surprising that we can use special forms of learning to create brain plasticity. There are also new forays in modulation, whether non-invasive or invasive forms like deep brain stimulation, although those are not yet ready for prime time for neuropsychiatric disorders, in contrast to movement disorders where DBS, there are probably 200,000 patients worldwide with DBS electrodes for Parkinson's. But the story of drug therapies, which are just going to remain critical, likely they'll be combined in the long run with psychotherapies, with neuromodulation. But from a global public health point of view, having pills in a bottle that can be distributed globally is really very important. But if you look, the molecular targets of every commonly used class of psychiatric drugs is the same as 1950s prototypes.
Can you use the mouse? It's a big fat
Is it? Well, let me. You know, I might not have the motor, you know, coordination. We'll see. You know, or I could, yeah. Or I might use the mouse and fall down. Yeah. So lithium, of course, is unique. Its therapeutic properties were identified first in 1949, observing guinea pigs. But given the way regulation worked in those days, it was pretty quickly brought into humans with bipolar disorder. All of the antipsychotic drugs have exactly the same molecular target as the very first serendipitously discovered drug, chlorpromazine, which is the D2 receptor. I mean, you know, there are other receptors that are targeted, but this is still the obligate molecular target. The antidepressants all target essentially monoamine reuptake or release. There's a new, you know, interesting observation that ketamine may be useful in acute depression, but that remains to be established. And then we have the benzodiazepines. And, you know, one question, again, for, you know, again, sort of a completely unfair, rapid overview of the history, but again, to introduce it to people who don't think about this every day, is the question of how did we get there, right? So basically, these drugs were discovered serendipitously. Here's a picture of Henri Laborie, a rather flamboyant French surgeon, who was using this drug chlorpromazine as a pre-anesthetic to sedate patients and control various autonomic functions. He was so impressed by the sedation that he called up his psychiatric colleagues, DeLay and Denneker, and said, why don't you try this on your psychotic patients? It turned out that the sedation was actually a side effect. There was tachyphylaxis to that. But over time, hallucinations and delusions started to melt away. So that was the discovery. The first monoamine oxidase inhibitor antidepressant was designed to treat tuberculosis. The mycobacteria were pretty unimpressed. But for those of you who've read, you know, The Magic Mountain, you know, there were all of these poor souls, ill, locked up in these sanatoria. Their moods brightened, and so, you know, the first antidepressant was discovered. Now, serendipity, of course, can be a wonderful thing. Here's Alexander Fleming, who discovered penicillin serendipitously, and nobody says, you know, what a pig, he let his agar plates get moldy, he should have thrown them out. No, you know, instead he saw these areas of clearing and recognized he had the first antibiotic. But the difference is that in microbiology, this serendipity led to a series of steps by which you could discover different kinds of antibiotics. You could plate different streptomyces, different bacterial strains, different molds, and use this kind of paradigm. Whereas for complex human brain disorders, you know, they're really, in fairness, in the 1950s, you know, nobody had any clue how you would go about discovering not just more of the same, but, you know, truly new compounds that would treat these terrible disorders, schizophrenia, bipolar disorder. And unfortunately, what happened was that people, first of all, they were very careful. They said, look, we know there's this post hoc ergo propter hoc fallacy, right? You know, which is just because aspirin relieves pain, it doesn't mean I had an aspirin deficiency. But what the heck, let's see what these drugs do to animals. And Dr. Portsault created this forced swim test where if you put a rodent in a beaker of cold water, it struggles, and then it floats. And if you give them an antidepressant, if you give them imipramine, the prototype compound, they struggle for longer. The hope was that there was something that was intrinsic to the disease process that was being treated by imipramine. The fear was that this was simply an assay to detect more compounds like imipramine. 60 years later, we know the answer, but that actually has not stopped people. Indeed, in I think really sort of an intellectually tragic move, behavioral pharmacologists started calling this behavioral despair. Whereas, you know, when the rodents stop struggling and start swimming, you know, I actually think the rodents who do that are pretty clever, right? They hydrolyze more ATP. Maybe this jerk will come and lift me out of the beaker. Carl Diceroth actually had the nicest explanation for this. He called it a transition from active to passive coping, as opposed to an animal model of depression, which it certainly isn't. And so this concept grew up with so-called predictive validity. That is, if a model or an assay was positive with an effective drug, then somehow this must be a disease model, which really was a self-deception. And then worse was this concept in this field of so-called face validity. Does the behavior remind us of the human symptoms? And so by calling this behavioral despair instead of a smart rodent saving energy, we began, unfortunately, a process of just got ourselves into a cul-de-sac, which is not to say there were a lot of really good alternatives. So what are the obstacles? Well, you could say a good sort of bioassay is the interest of the pharmaceutical industry. So these illnesses are really common, schizophrenia, bipolar, autism, all 1% of the population, it adds up. Major depression, 15 to 20% of the population. Severe burden, as I showed you in the first slide. And not only do we have only the same molecular targets, but efficacy actually stalled. That is, the latest, greatest antidepressant that you can now buy is no more efficacious than imipramine was in 1957. It's just safer and more tolerable. So pharma has exited. They've said, well, you don't have any new molecular targets. And then I think this is really, in some ways, the biggest problem is, cancer is a hard problem, but it's an easier hard problem. A surgeon hands a scientist the disease. You do an excisional biopsy, and there's the disease, right? Whereas this is from an Inca trephination. This is now considered sort of impolite. You can't sneak up on somebody and take a few cells. But even if you could, cancer is much more cell autonomous, and our diseases are diseases of circuitry, right? So in the sense of what's proximate to the symptoms and the impairment are failures of circuitry. So we have this really difficult issue of, what are the tissue, what are the models in which we're going to study these diseases? Now, the other thing about the human brain, of course, especially the cerebral cortex and most especially the prefrontal cortex, this is very recent in evolution. And so it is very challenging to model a lot of what we're interested in traditional animal models. One exception is, there are some very rare penetrant monogenic forms of autism and intellectual disability where you can at least make a transgenic mouse. And if you focus on brain structures that are lower in the neuroaxis, that are evolutionarily older, you can learn a lot. But we really don't have, in our widely used rodent models, it's hard to argue that the prefrontal cortex and the neuroscientists know how different they are between, rodents and humans that have different cells, different circuits. In a nocturnal animal where their evolutionary pressures were very different than in a diurnal visual animal like us. And we have only descriptive diagnoses. This is, so there are no biomarkers for clinical trials. So the industry, despite the enormous need, has basically said, we don't know how to get from here to there. We're kind of out of here. All right, so one thing we always knew was that there were molecular clues to pathogenesis hiding in our genomes because in the interest of time, I won't take you through this one, but the heritabilities calculated from twins, which are estimates of autism, schizophrenia, and bipolar, are among the highest of any sort of relatively common human complex disorders. So in the range of between 65 and 80%. Major depression, in contrast, which is much more heterogeneous, much more common, has a lower heritability and is a much harder problem. So, for something to be heritable like this, it means that genetic variants, in your brain are influencing the likelihood of disease. And of course, genes are not fate, but they play a very big influence here. The problem is, our brains are not like Mendel's peas, right? The determinants of these turn out to be many, many hundreds of genes and thousands of different alleles in different combinations that create disease risk. So really until about 2005, while this is the number of human traits, these are Mendelian traits being solved, increased enormously for polygenic or complex traits just in animal models, especially things like Drosophila, some rodent QTL studies, there was a little bit of progress, but in human complex traits, really nothing. And just autobiographically, since Bill mentioned it, when I left NIMH in 2001, I decided I didn't want to do the kind of basic molecular neurobiology I was doing anymore. I was really interested in the diseases. But I was in such despair. I mean, we knew the genetics would eventually work, but we just had no idea how to make it work. And it seemed like there were no other real clues given the lack of models that I said, what the heck, I'll sort of take a time out and be Harvard provost for a few years. Little did I know that that was not going to be the most fun you could have. But in the interim, what's happened is technology. So, we scientists always like to think we're smart. There's a painting of Galileo, and they always paint his forehead as gleaming to show his intelligence. But down here in the dark is a telescope, right? And it was, after all, incremental advances in grinding lenses that allowed him to build a telescope and make these new observations that were so important. So what we need is new observations, right? Psychiatry has been living on the serendipitous findings. What is the mechanism? And so we've had dopamine hypotheses. People have been living on refried serotonin. But we need a whole set of new observations to empower, a whole new set of hypotheses. So since the Broad is technology driven, but also given this belief that we are in a phase where we need to be much more initially Baconian, and then we can have more traditional hypotheses again, I've tried to, and as you'll see, really push the genetics, but also as we interrogate the genetic results, remain unbiased and high throughput for as long in the process as we can, to recognize that the more traditional biochemical methods where you grind up the brain, and spin out the skull and the hair and do assays on the supernatant is not going to tell us about how the nervous system works. So we have to really respect the challenges posed by cellular diversity, but these are really hard, and really think about the right human cell types. So what have we been doing? Well, the first thing is, at the time when I returned to the Broad, the technology has, as I've already implied, really turned the tide. The cost of sequencing DNA has come down, depending on how you calculate it, about 1 million-fold in the last decade. It makes Moore's law look kind of torpid. In fact, right now, even if I'm being a good accountant and include overhead, I'd get a whole genome for less than $2,000. But I pay Google or Amazon $500 for the data transfer and storage. So these costs are beginning to converge. And of course, microarrays, gene chips where various gene sequences are etched and can be assayed very simply, are also really powerful. I'll talk about it in a bit, but there's a psych chip that has a good common variant, common genetic variant component and a rare variant component, and then the whole community got to nominate 50,000, copy number variants and other things that have been implicated in psychiatric disorders. That costs $70 a person, to do that, and the next generation we think will cost $50 a person. So you can actually now begin to study lots of people. Now, just in introducing this, it's useful to think in terms of human population history. So this is just world population. This is years before the present, starting at, let's guess, 100,000 years. And you can see that humans perked along at some tens of millions of people on the planet for a long time. Then we had the Neolithic Revolution and then the Industrial Revolution, and now we have 8 billion and going. And this has happened only in about 12 or 15 generations. So no time for cleansing natural selection, and there are about 60 new variants per meiosis. And so there are lots and lots and lots of neutral and interesting variants in all of our genomes, and most of us differ by at least 1 million base pairs, even if we feel like we're quite phenotypically similar, being academics and so forth. But this ancient variation, which has had a long time for natural selection, it creates a lot of common variants, and that's what can go on a chip. But to study more recent variation, that you have to sequence for. And most of the focus still is on the protein coding parts of the genome, because we understand it, and frankly, it's only 1% to 2% of the genome, so it's a lot cheaper in every way, about $300, and the data storage is not such a problem. So using these technologies, I think we can skip this. There's one other thing that, for the biologist, is very disconcerting, which is, as I'm going to show you, we're discovering lots and lots of alleles, flavors of genes that contribute to risk, but they almost invariably have a small effect size. And people keep looking and looking for, rare alleles of large effect. And again, in autism and intellectual disability, there are at least a few of these. But here's an analysis that was done there have been a number of these done in Scandinavia that looks at the fecundity of people. That is, you compare someone affected by schizophrenia or autism or bipolar disorder with their unaffected sibling and ask, on average, how many children do they have? And to just summarize the most salient points, and by the way, males have an earlier onset of schizophrenia and tend to have a worse course. So a male with schizophrenia has about a quarter of the number of children that their unaffected sib has, and a female with schizophrenia, maybe half as many. Male with autism, also about a quarter of the children. And the kids with these very severe single gene variants that cause autism and intellectual disability are not always, but tend to be very impaired and tend not to transmit these alleles, many of which occur de novo. So sparing you the calculation, Mark Daly and Ben Neal have looked at what strength of allele, what odds ratios can actually survive these really pretty rigorous selection processes. And it turns out that any allele, that is any flavor of a gene, that increases the risk of schizophrenia or autism by more than 1.1-fold is quickly swept out of the population. So there's a ceiling on the biological effects of the kinds of genes we're finding, which creates enormous challenges for this community. This analysis, of course, has come recently. Initially, people got these new technologies, realized that the older methods that had posited genes of large effect had failed, and got what they thought was a large sample. And it was at the time 2,600 cases, 3,300 controls. And this is called a Manhattan plot, although this obviously looks more like Manhattan, Kansas. They're supposed to be skyscrapers. So this is just the -10 log p, which is the giving you a measure of whether the results are likely to be true. This conventional line of significance is just p of 0.05 corrected for 1 million comparisons. And these are your chromosomes arrayed at the bottom. And at this point, there were 0. And at this point, also the NIH peer review system told, I was still laboring away in the Harvard provost's office, but told my future colleagues, well, you tried it, it failed, it's over. But in fact, there were all kinds of reasons to suspect that it was just underpowered. And one was that if you just took these hits and looked at, sort of made a function and created what's called a polygenic risk score, and looked at the 10% of people who had the greatest pileup of even these sub-threshold findings, they had a much elevated risk, say, of schizophrenia, or same thing for bipolar disorder. So that suggested that in this noise, there really were some true findings. And so in 2011, with now 21,000 subjects total, all of a sudden there are 5 genome-wide significant hits. And in last year, at the last data freeze that's been published, with 37,000 cases and 113,000 controls. And this, again, is with a common variant chip. These are all common variants. There are 108 independent loci. And right now, actually, you know, the numbers are going up, and they're about 130. And we're still on the steep part of the discovery curve. Now, I would emphasize that the odds ratio conferred by the average one of these is only 1.08, an 8% increase in risk. So for biologists, you know, this is a very big challenge. I would also just point out that the biggest risk by far is in the MHC locus. In fact, that association is so strong that it was known in the pre-DNA era, when we only had ABO blood groups and some HLA antigens. So I'll come back to that in just a moment. The other thing about these common variants is that they are 90% across all human diseases regulatory. You know, evolution has maybe a little bit, you're less likely to be selected out if you have a new variant in a gene regulation region. Certainly, what you see in terms of evolutionary conservation is that the DNA sequence and the protein sequence of proteins tends to be conserved. And just to illustrate the kind of finding, so there's a calcium channel subunit, CACNA1C, which is the alpha 1C subunit of an L-type calcium channel. And there's a toxic gain of function mutation that causes Timothy syndrome, which has mental retardation and autism and facial dysmorphology, and not surprisingly for calcium channel, cardiac arrhythmias, and is often lethal early on. The very same gene is implicated in schizophrenia and autism, and this is one of the variants that has been fine mapped, and it maps to an enhancer within an intron, and preliminarily it appears to decrease expression by, you know, 5 or 8%, maybe slightly changing the stoichiometry of assembly of calcium channels in those cells in which that enhancer is used. So a much more subtle and challenging kind of deficit. So those are common variants. And then, of course, we've been sequencing to find rare variants. So there are two designs when you're looking for rare protein-altering variants. I should have said, I'm sorry I didn't, that in looking at common variants, these are all case control. That was probably implicit. Well, but one is to look to unaffected parents and an affected offspring, so-called trio design, and sequence everybody and look for new mutations that might be disease-associated. And most of these turn out, of course, to be red herrings. But in the case of autism, there are a number of really interesting de novos that seem to be associated with, again, autism, but associated with severe intellectual disability, greater risk of seizures. These include SYNGAP1, an important gene involved in synaptic plasticity. You'll recognize a sodium channel gene. And many of these genes with different flavors, different alleles, are also found in intellectual disability searches, in autism searches. So this has been very fruitful. And of course, these de novos are highly penetrant, right? Parents were healthy. They have to have a certain genetic background, but they cause disease. And here you can make transgenic, mice, zebrafish, you name it, and learn important things, as long, again, as you're thinking about brain regions that are conserved in evolution. And I'm not going to go into the detail. I think we can miss that. And then there are the case control. And here, we were very hopeful at first that, these are relatively recent in evolution, right? We've had this vast population expansion, and maybe some of them would have greater effect size. But again, the fecundity analysis that I showed you would predict that that hope was in vain. And in fact, right now, there are 6,000 sequenced schizophrenia cases and, and a much greater number of controls from across many different disease studies. And not a single gene is yet positive. That is, it turns out that the rare variants are both of small effect, and they're so rare that you just can't get a p-value. And in many cases, we may run out of humans before we get a p-value for these things. And so, again, in a careful way, rigorously trying to avoid self-deception, we're going to have to bring statistical genetics and biology together to try to figure out what some of these things are doing. Now, what we can do, of course, and what we will do, is vastly increase the sample size. We can focus on those genes where there's interesting protein coding changes, where we already know by common variant, by GWAS analysis, that the gene is significant, and we can ask whether the effects are convergent or divergent, and somehow think about that. Some people have simply focused on the trio studies for kinds of variation that we understand, like loss of function. But this is going to be an enormous challenge. So we have been the recipients of really astonishing philanthropy, and my view is that we should, work to push along all of the biology. But the most important thing we can do is, with respect to genetics, is literally to kill the problem and make everything public. And so with lots of collaborators around the world, starting with the Psychiatric Genomics Consortium, we'd like to, in the next 7 or 8 years, sort of analyze the most heritable diseases in psychiatry to diminishing returns. And I think we'll know what that looks like when we hit it. So just to give you some sense of the numbers in schizophrenia right now, there are actually 60,000 cases. As you know, we've sequenced 6,000 exomes, 1,000 whole genomes. And in the next 3 years, we're hoping to have it more than 100 common variant, GWASs, 40,000 patients with plus controls, exome sequencing, and 10,000 whole genomes. Autism is lagging a bit, but this is just to give you a flavor, and we're always trying to see whether we can find more collaborators and more funding, because this is sort of the grist or the platform from which biologists, computer scientists, modelers can begin to make inroads. And the other thing is we recognize that initially we had started with convenience populations, that is all Northern Europeans, because of these amazing Scandinavian population registries and these well-organized health systems. But that was just a tiny sliver of human genetic diversity. And so we've begun collaborations now in Japan, in Shanghai, and in Beijing, in Mexico, but looking, and other places in South America, but looking for the non-Euro part of the admixed populations. And very ambitious, but I think for global equity reasons, but also because of the remarkable genetic diversity in sub-Saharan Africa, we're beginning collaborations. The only place with the infrastructure right now is in South Africa, and we're working with the Wellcome Trust actually to create sort of training programs for investigators there, mindful that we don't want to create a brain drain, so we want to do it there. And this will be a pretty challenging, but I think really important aspect of this discovery. If these diseases stratify, for example, we want to make sure we actually don't miss whole chunks of human population. So people ask, okay, great, you said all this, but why do you care about alleles of small effect? So I think the issue is what's important, no matter what the effect size is, if you're confident, right, if you've achieved real statistical confidence, then any allele is a pointer at a gene. It's saying the gene I reside in is involved in the disease process. And of course, genes then point to pathways and molecular complexes, although easier said than done because of the cellular diversity in the brain. And so the notion is one of the reasons to, genotype and sequence to diminishing returns is not, again, not just to end up with the, mother of all gene lists, to rephrase an old Saddam Husseinism, but to look for the convergence of this genetic information. And the hypothesis, based on other diseases, including cancer, that have been studied, is that these many, many hundreds of genes, maybe more than 5% of the genome, are going to converge on a finite number of biological pathways. Now, we don't know that for sure, but there's already some evidence of that. The trouble with finding biological pathways in the brain, if they're hepatologists here, I don't mean to denigrate your favorite organ, but there are hepatocytes and Kupffer cells and endothelial cells, and we have a still unknown but many thousands of different cell types. And so single cell analysis is really critical to figuring out what the cell types are. And now that there are, again, because of time, I'm not going to take you through. You could look at Evan McCosco and Steve McCarroll's recent Cell paper, but they're really good ways of doing single cell transcriptomics now. Now let's see if I can. I'm not sure I can start the video here. Let's see. No. It's not. This is just a nano machine that Ani Basu created in Dave Waits' lab. They work on droplets. And basically what it does is it brings barcoded beads with a poly-A tail together with thousands of individual cells, puts them in a droplet, uses basically turbulence to lyse the cells, and then you can do massively parallel RNA-seq, and they've done experiments with now tens of thousands of cells pretty quickly. So we can, and then because of the barcoding, you can figure out which RNAs came from which cells. But this is the kind of thing, we can't just, again, we can't, in the nervous system, we can't find pathways by, grinding tissues up. The other thing we don't have, of course, that we're really going to need, because the targets for therapeutics are actually proteins and protein complexes, is we don't have single-cell proteomics. This is a watch parts. We want to be able to figure out how it comes together. And here, it's really difficult. Any brain dissection, no matter how careful, has many cells. So one of the things we're doing is making industrial quantities of certain, stem cell-derived neurons. We've hired our first process engineer. And what we are doing is interaction proteomics in uniform cell types, and then trying to go back to human postmortem brain to check our work. But I will confess, it's a really inelegant, iterative process, but we don't know what else we can do to find these protein complexes in which risk genes find. Now, we've had some luck. One is that big signal in the MHC locus. Turns out, and here he's talked about it, even though the paper is still, it's in revision from Steve McCarroll and Beth Stevens and their groups. It turns out that within the MHC locus, which is devilishly difficult, the signal was coming from complement factor C4. And when Beth was a postdoc here with Ben Barra, she did an unbiased screen and found that complement was the eat me signal in, actually that was C3, but it's all part of this cascade, in synaptic pruning. And because schizophrenia is a disorder in which in periods of adolescent synaptic pruning, there appears to be severe overpruning, loss of synapses, loss of dendrites that is accompanied by cognitive decline. This single GWAS hit, which increases risk only by 10% if you have the worst allele, points to a biological process which is plausibly involved in schizophrenia and allows us to begin to do this work. I'm going to slip over another example and just say, so here's another Stanford picture. Clearly, we love mice, right? We can do all these amazing things in mice. The trouble is they are low throughput. How are we going to interrogate the function of hundreds of genes, thousands of alleles? If you put even the most penetrant of the common alleles, 10% increase in risk, in a mouse, the genetic background is going to dominate, right? In fact, by definition, for these polygenic diseases, what matters is genetic background, right? It's the collection of all of the genes that come together to increase risk. And we could say, well, you can take one of these genes and make a knockout, but if you just think about it, that's not going to help you. One of the reasons I showed the Timothy syndrome slide is you can have a gain of function or you can knock that gene out, but that's not going to tell you what a small difference in expression or protein structure in 100 brain cell types is doing to the phenotype. So we really have a lot of work to do. And just sort of to end, I think we have to spend more time in neuroscience than we have in designing models, thinking about evolution. In basic science, you're unconstrained, right? I mean, you can take any model that's informative and derive general principles, and that's the right way to do it. But if you want to do something translational, the model needs conservation. If you're going to make a drug, what has to be conserved is not, you can't say, well, mice have conditioned, fear and humans have conditioned fear. The question is, if I'm going to target some molecular target in the brain with a medicine, is that molecular complex and the cell type and the circuit conserved between my model and the human so that I get the output that I expect? And to date, people have not been thinking that way. And so we have to match our models to the task. Right now, people are appropriately doing, iPS cells in culture, cortical spheroids, really interesting work with organoids that lots of groups are doing, which is still early, but will give us sort of mini brains, just illustrating the cool things that organoids give you, doing drop-seq with organoids. But to end, we are going to, I believe we're going to need all of this genetic information to make it available to everybody. We need molecular tools at the single cell level to understand how these genes are acting. And maybe the hardest problem for this community is to figure out what models to test hypotheses in. And yes, mice and stem cells and organoids. There's now a new effort that we're involved in that Guoping Feng is heading to make CRISPR-Cas9 engineered marmosets with some of these genes. But ultimately, I think we finally have the molecular clues we want. They just haven't come in a form that makes it easy. This isn't cancer with highly penetrant somatic mutations. This is a very different beast. Thank you.
So we have time for a few questions, and we have runners with microphones. There's a question right here, right in the middle.
Fantastic talk, Steve. Thank you. I want to raise two related kind of questions and have you give your thoughts. One is the role of brain imaging. And the second, which is a complement, which is that through a lot of brain imaging now, when you start to look across many disorders, it's coming out more and more clearly that there's a lot of commonalities in terms of types of brain circuits and what effect that has on the genetics.
So very quick answers. The first thing is that, in cognitive neuroscience with well-chosen hypotheses, brain imaging has been, I think, remarkably informative. I'm talking about functional imaging. In looking at disease, I actually think that as grants leave the NIH, they are largely stillborn because they're underpowered given the heterogeneity of these diseases. And that's an issue that it's very hard to solve because everybody wants more grants, but the heterogeneity is really daunting. There's this Enigma consortium trying to make sure that through meta-analysis some of that can be overcome. That's problem number one. Problem number two are the disease definitions. So what we're doing in genetics, I didn't say this, all of our new cohorts are recontactable because it turns out that if you really deeply phenotype somebody on the way in, all you do is decrease your power. What we want to do is recall people by genotype and come back with very new phenotypic hypotheses, which might include imaging. But I think, again, if you're looking for biomarkers which can be broadly used, I think physiology, in some ways is even more promising because more portable and less expensive potentially. But it's a brief answer to an important question.
I think over here, Jennifer.
I guess a difference between cancer and the brain diseases is that cancer, we know what the common attribute is, right? It's unregulated growth.
Yeah.
And in
you have a great
brain
assay.
diseases, what if the common attribute is at the circuit level? What is the path to get from all of these genetic variants to that common attribute?
Right. So that's why the models are so, I front-loaded this to show you the progress in genetics and where we're going. But that's, so clearly IPS cells that are dissociated don't tell you about circuits or even synapses in a really veridical kind of way. I think the organoids could be promising, but clearly at some point we're going to need a living organism, and in some cases I think it's going to have to be a primate, frankly. But we also have to think more about the human model for the human, which gets us actually back to physiology and imaging and finding ethical and pragmatic ways to understand human circuitry. Very quickly, because evolution really in some ways solved, fear circuitry and reward circuitry pretty early. If you're a freely living critter, those things have to be intact. But that doesn't teach us about the diseases, because the problem in humans is actually prefrontal cognitive, top-down control of excessive or inappropriate fear or reward. And so we're not going to, we're not easily going to model that. Maybe in primates, but maybe not. Bill seems to think that their temporal discounting is imperfect, a little impulsive, like some colleagues we have. Yeah. So we're going to use those, but I think we have to really think about human models for the human as well.
Comment on the epigenetic landscape as well as microRNAs.
Yeah, well, so I wish I could. Epigenetics is exquisitely cell type specific, and just as we are trying to use IPS models to do, proteomics and single cell analyses, we also need them to do epigenomes, again with the recognition that growing up in an incubator is somewhat different from growing up in a brain circuit, and so we're always going to have to be iterating between postmortem human brain and these cellular models. But right now, we can't do the kind of unbiased epigenomics that we really are going to have to do.
So I would like to thank Steve for this spectacular overview. It's sobering, but to me, it's wonderful that there are leaders like Steve who are just taking the science and saying, where can we do this, and thinking reasonably and rationally in a hard-nosed way about it. And I think this is the way forward. And Steve, thank you so much.
Great pleasure. Great to be here.
For more, please visit us at stanford.edu.